Oke, kita mulai. Yuk, kita mulai.
Baik. Good morning, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Welcome to webinar series on applied microbiology BRIN with topic sustainable innovations, harnessing nanoparticles for nanobiotechnology and microbiology, held by Pusat Center for Applied Microbiology, National Research and Innovation Agency BRIN today, Wednesday, March 5th, 2025. In this occasion, we are honored to welcome Head of Research Center for Applied Microbiology BRIN, Dr. Ahmad Fathoni, our speaker, Syed Tawab Syah, Ph.D., a postdoctoral of Research Center for Applied Microbiology BRIN, and our moderator, Puspita Sari, Ph.D., researcher of Research Center for Applied Microbiology BRIN. Ladies and gentlemen, let's start the agenda with opening remark from Head of Research Center for Applied Microbiology BRIN to Dr. Ahmad Fathoni. Time is yours.
Okay, thank you, Ms. Utsi. Assalamualaikum warahmatullahi wabarakatuh. Very good morning, everyone. Honorable Speaker, Dr. Tawab Syah, our postdoc at Research Center for Applied Microbiology, with Dr. Puspita as a host, and everyone who has joined our webinar today from Research Center for Applied Microbiology and also other research center and maybe also from university. First of all, I would like to thank you everyone for joining us this morning, and also thank you very much for Dr. Tawab for accepting our invitation, and he's going to share his long experience in nanoparticle research. And yeah, this is very interesting topic because currently we know that we are talking about nanotechnology. So this is like nanotechnology has experienced a rapid growth and source of promising potential in various fields, including microbiology. Because we are here from applied microbiology, then I would like to focus on microbiology or maybe in a biotechnology field as well. And why nanoparticle? Because nanoparticle with their incredibly small size, have demonstrated the ability to enhance the efficiency and effectiveness in application. So using nanoparticle in microbiology application, we hope that we can develop new product based on microbes, or it can be enzyme, it can be metabolite or microbe itself, using nanoparticle, and then to enhance their application. So Dr. Tawa will explain more about it, and also maybe he will also explain a bit about his research in BRIN with Dr. Ira. He has, and with Dr. Ira, have created several compound actually using nanoparticle, which is interesting. And currently he has promising also result, and one paper accepted and then 3 papers submitted, which is great to me, and he has been contributing a lot in our center, and hopefully we can extend this collaboration with him after he finishes postdoc, and then in the future we can still collaborate and also even expand collaboration. So if participants here interested in his topic, you can contact him, and then we can expand our collaboration, more collaborator, more research, and I'm sure that we can get a faster result, output, and greater impact. So let's do this webinar this morning, and hope you gain more knowledge from Dr. Tawab experience and also research experience. Okay, I think that's all from me. Thank you very much, everyone, and hope this webinar goes smoothly as planned. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Fathoni. Ladies and gentlemen, before we start the agenda, we will have a photo session first. To all participants, please activate your camera and for the committee to take the screenshot. I will start count from first slide. Ready? 1, 2, 3. Second slide. 1, 2, 3. Third slide. 1, 2, 3. Thank you. And I would like to remind to all participants, please fill the attendance form from the link provided by the committee in the chat box. Now we are going to start presentation and discussion session, which will be led by Ms. Ira Puspita Sari. Ms. Ira, time is yours.
Thank you, Ms. Arini. Can you hear my sound, okay?
Yes.
Good morning, everybody. Good morning, everyone. Welcome to our event today. It's wonderful to see so many enthusiasts. Until now, there are 50% joining at this moment. I'm Ira Puspita Sari, and I will be your moderator for this session. But first, I want to share the curriculum vitae of Dr. Tawab. Okay. Can you see, audience? Yes. Dr. Tawab, his full name is Syed Tawab Syah, Ph.D. This is his email. He has many work experience. He finished his Doctoral Master Science Chemistry and M.Phil Chemistry in Pakistan, Hazara University and University of Peshawar University, Pakistan. And for his postdoc Ph.D. Chemistry in University of Malaya, Kuala Lumpur, Malaysia. And now he is as a postdoctoral research fellow at Applied Microbiology BRIN. He has many publications, but we saw only a little. His Google high index corpus is 7 with many citations, more than 200. And his citation in Google Scholar is more than 600. His area research is on many things, but now he's doing with us on biogenic nanoparticles and organic nanoparticles. Using synthesis using bacteria microbiology. So that's all the CV, the curriculum vitae. So I don't want to talk too much. Now the time for Dr. Tawab. Time for you, Dr.
Good morning, everyone. Assalamu alaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you, Pak Fathoni and Dr. Ira. So today, I'm Sahabat Iptek, and I'm excited to share my research on sustainable innovations using nanoparticles and biomaterials. Today, we will explore how these tiny tools can revolutionize nanobiotechnology and microbiology for a greener future. So I think no need to discuss more about myself, as Dr. Ira already introduced me to you people. My research area mainly focuses on the intersection of chemistry, material science, with a strong emphasis on developing sustainable solutions. My expertise in nanoparticle synthesis and functionalization has led to numerous publications and ongoing. There are several ongoing projects in BRIN in which I am actively involved. So today we will. My title is Sustainable Innovations Harnessing Nanoparticles for Nanobiotechnology and Microbiology. It is a path towards a greener solution to the many problems in the world. So let's start with the basics of nanoparticles. Nanoparticles are incredibly small particles, but their unique properties make them powerful. Their high surface area allow for enhanced interaction, leading to remarkable applications in numerous areas. In this figure 1, you can see the comparison of sizes of different mm, materials. So 1 nanometer is one.
Excuse me, doctor.
Yeah.
The skin there is no the share screen, please.
Oh, there is no share screen?
Yes, can you share the screen?
Okay, I will do it again. Can you see it now?
Yes, full screen, please. Yes, thank you.
So should I start from the beginning?
Yes, yes.
Okay. So good morning, everyone. I am Syed and I'm excited to share my research on sustainable innovations using nanoparticles and biomaterials. Today, we will explore how these tiny tools can revolutionize nanobiotechnology and microbiology for a greener future. Today, I will also discuss some general applications, which is not our research, but I will talk about it in general. So let's start with the basics of nanotechnology. Nanoparticles are incredibly small, but their unique properties make them powerful. Their high surface area allow for enhanced interactions, leading to a remarkable applications in numerous areas. According to definition, nanomaterial is material which the size ranges from 1 to 100 nanometer, either in the form of cluster, agglomerate, or a single nanoparticle. And these are collectively called nanomaterials. And 1 nanometer is 1 billionth of a meter. It is extremely small material. In figure 1, you can see the comparison of different materials. Like, for example, our human hair is 100,000 nanometer, while bacterial cells are 10,000 nanometers, while our RBCs are 7,000 nanometers. Fungal species have a size of 500 nanometers. Antibodies have size of 10 nanometers. Water molecule has particle size of 0.3 nanometer. And this is the scale range. So I think we are aware of the size of nanomaterial. So I will go to the next slide. So I will start with the history of nanomaterials. The term nanotechnology was coined in 1960s, which is infant field compared to other fields like chemistry and physics. But today we will talk about we will explore about the intersection of art, science, and evolution of nanoparticles through the history. In this figure, you can see that this is a time scale which is starting from Big Bang and then moving to the 21st century. Today, we are consciously harnessing the power of nanoparticles in countless applications from medicine to electronics. This timeline underscores the enduring relationship between art, science, nanoscale, and reminding us that even the most cutting-edge technologies have roots in our ancient past. So this is a very interesting slide. Figure 3A, here you can see a painting, which is original painting of a Bobby Research. Sorry if I'm not pronouncing this correctly. It's like big deer from Indonesian island of Sulawesi. And these are the prehistoric caves paintings typically contain like natural pigments derived from minerals like iron oxide, which naturally contain nanoparticles. And these nanoparticles in the pigment contribute to the color and longevity of the artwork. Although prehistoric artists didn't have a scientific understanding of nanomaterials, but they were effectively utilizing nanomaterials that contain nanoparticles. So our ancestors, our forebears, used these carbon nanostructures and
ya, sebentar ya. Bapak Ibu, apakah bisa ada yang terputus ya ini ya? Sebentar Bapak Ibu ya, sedang di-connect dengan mobile data karena koneksi di BRIN sedang tidak baik-baik saja.
Hello?
Yes, please, Doctor, share the screen first.
Yeah, sorry for the inconvenience. Can you see my screen? Can you see my share screen?
Okay, we can see.
Yes, I can see.
So this is the image of the original painting of dari Visa, which is a pig deer from the Indonesian island of Sulawesi. And on the right side, the images I have captured in a BRIN office. I think BRIN is also actively working. BRIN researchers are working on these old days caves and on the earth also. And this is also very interesting that these are the first in history of the painting. So Lucas, you can see it here, and it is showing like two different colors, like green and red, due to presence of silver and gold nanoparticles in certain ratios. Similarly, image on the right side, left side, is showing the particle sizes of different paints and pigments used by ancient artists in different, like for example, glazing the pottery or for the glass, like in the churches there are like colorful glass. So they were using these tinted colors, but they were not aware of the term nanotechnology, but they were using it in the art. So this not only showcases their skills, but also underscores the long history of human interaction with nanomaterials. And this slide illustrates that manipulation of materials at nanoscale is not modern innovation. Ancient civilizations, specifically Egyptians, were creating and utilizing nanomaterials thousands of years ago without our modern understanding of nanotechnology. And it also reinforced the idea that nanotechnology, while a modern field of study, has roots that extend into our past. This figure A is a picture showing the hair dye. The Egyptian, they were using hair dye, which was in fact nanomaterial. Produced from those were like plug-in sulfide nanoparticles, and the image B is showing like the paintings. They were using blue color, which was calcium sulfur silicate and silica nanosheets of less than 5 nanometer thickness. And the remaining images are showing their SEM and TEM images. So sustainable nanoparticle synthesis are green methods, which is our main concern in BRIN, and also it is globally people are doing these methods. And here we are focusing on biogenic synthesis of nanoparticles for reduced toxicity and energy use and biodegradable materials. Sustainability is the key. We focus on the green synthesis methods, which minimize environmental impact by using biological agents and reducing the toxic waste. So Here I will start with the zinc oxide nanoparticle synthesis and its applications. This is the conventional method which we have used in our lab. So zinc oxide nanoparticles are dual action to threat to bacteria and cancer cells. So zinc oxide nanoparticles are fascinating class of materials with wide range of applications in nanomedicine. They show a great promise due to their unique properties. Their dual antibacterial and anti-cancer activity makes them particularly attractive for developing multifunctional therapeutic agents. So zinc oxide nanostructures have ability to form ROS formation, which are reactive oxygen species, and also they produce zinc ions, and then they have interesting surface chemistry. We can functionalize them with different with various molecules, and then easy or facile synthesis methods, and also variety of shapes like nano sheets, nano spheres, nano rods, and nano flowers, depending on the synthesis method. And then it also has luminescent or optical properties, and these are the properties which we manipulate in our application parts, such as anti-tumor, diabetes treatment, antibacterial, wound healing, antifungal, and anti-inflammatory. So what is the mechanism? How do nanoparticles achieve their dual action? Antibacterial, in case of antibacterial activity, they generate reactive oxygen species causing oxidative stress and bacterial death and disrupt the cell wall membranes. In case of anti-cancer activity, they cause ROS-mediated damage, including DNA damage, and disrupt signaling pathways. So zinc oxide nanoparticles achieve this dual action. They generate reactive oxygen species. Like you can see here, like zinc oxide nanoparticles, they produce reactive oxygen species which cause oxidative stress and causing the damage to the cell. And then second mechanism is absorption on the surface of cell and disrupting the cell membrane or cell wall. And then the third mechanism is the zinc oxide nanoparticles produce zinc ions, which also interact with the cell membrane and cause damage to it. So by carefully engineering the surface and composition of zinc oxide nanoparticles, we can tailor their properties to achieve specific biomedical goals, paving the way for effective therapies. For example, if zinc oxide nanoparticles, if the surface charge is negatively charged, we can functionalize the nanoparticles with various molecules, so which can give the surface charge either positive or negative. So if they are negatively charged, they have antibacterial and anti-cancer activities due to ROS generation, as I discussed earlier. And if they are positively charged, then they can release the Zn plus R2 ions, like Zn plus ions, which also can damage the, which is main cause, one of the main cause of the these activities like anti-cancer and anti-bacterial. So this is an overview of conventional synthesis methods of zinc oxide nanoparticles. Usually we take zinc salt or zinc precursor, for example, zinc sulfate or zinc chloride or zinc acetate, and then we use any reducing agent Like in this case, we have used sodium hydroxide, and then we reduced the zinc precursor or zinc salt, and then we got like zinc oxide precipitates, and then those precipitates were centrifuged and then washed several times and dried in the oven. And this figure is showing like how nanoparticles are formed in the flask. Like nanoparticles are formed and then they agglomerate together. I will explain in the upcoming slides. I will explain more about it. So these are the characterizations we have done: FTIR, Raman, and UV visible. Confirm the formation of zinc oxide structures. And then in the next slide, I will show FESEM and HIPEM results. And we could get the particle size, average particle size of 3.5 nanometers. Mainly it was spherical shape. And then EDS also confirmed its EDS is actually elemental analysis. It confirmed the presence of zinc and oxygen. So here are HRPEM, SEM, and EDS images. On the left side, there are the three images are FESEM images of our conventional zinc oxide nanoparticles. In the middle, the images are from HRPEM, and this figure E is showing the average particle size of our nanomaterial, while figure F is the SAED selected area electron dissection, which also help us to identify characterize sample. And this EDS figure A is like you can say like a PEM image of our nanoparticle. Figure B is a overall image of it is like elemental mapping. So figure C is showing like the purple color is showing the zinc presence of zinc, while green color is showing the presence of oxygen. And the down there is the EDS spectra, which is also confirming the presence of zinc and oxygen in our nanoparticles.
Excuse me, doctor, we cannot see the screen. How about others?
Can you see the screen,
Yes,
participants?
yeah, I can see the screen.
Okay, should I continue or share again?
Yes, continue, doctor, please.
Okay. So we tested antibacterial activity using agar-well diffusion method. Zinc oxide nanoparticles showed significant inhibition against both E. coli and S. aureus, with E. coli showing better susceptibility, likely due to cell wall difference between gram-positive and gram-negative bacteria. And then we also evaluated the anti-cancer activity against MDA, MD, 231 cells using entity assay. We observed concentration-dependent cytotoxicity with an IC50 of 106 microgram per ml, suggesting cytotoxic affected likely mediated by ROS. I will skip this slide because I think my presentation is so long. So the second work I am presenting, which we have done in our lab, is biogenic synthesis of nanoparticles, a sustainable solution for antibacterial applications. Here, I am not discussing about anti-cancer because we are still in process of testing for anti-cancer. Some of our products are tested, some are ongoing. So zinc oxide nanoparticles have wide range of application, as I discussed earlier. We use like different methods for the synthesis, like just now I discussed about conventional method, which is a chemical method. There are also various physical methods to synthesize nanoparticles. So approach is both top down and bottom up. But these conventional synthesis methods, either physical methods or chemical methods, we need harsh conditions. Harsh conditions means, for example, for physical methods, we need high energy, while for chemical methods, we use like toxic reagents and harsh conditions, which can adversely affect the environment and human health. These methods are often expensive and require high temperature pressure, which contribute to the generation of hazardous byproducts. While biogenic synthesis using microorganisms or plant offer a sustainable method for the synthesis, and this method has gained popularity to address these issues. These methods employ plant extract, fungi, other biological sources to produce nanoparticles, which are more eco-friendly. So here is the overview of the one of our paper, which is Nanofactory, Biogenic Synthesis of Metal Nanoparticles for Sustainable Technologies. So this slide illustrates the concept of biogenic synthesis, a sustainable and eco-friendly approach producing nanoparticles. We are essentially using nature's nanofactory to create valuable materials. Let's walk through the process of how this happens. We start with a microbial culture. Let's say here you can see in the first class on the left side. These microorganisms or their extract act as a reducing agents. In case of conventional method, we use like any reducing agent, for example, sodium hydroxide or ammonia. But in case of biogenic synthesis, we use either plant extract, which act as a bioreductant, or we use any microorganism or their extract. Extract means in case of plants, those are extract, but in case of microorganisms, those are like their metabolites, secondary metabolites or primary metabolites. So these microorganisms are their extracts can act as a reducing agent or a bioreductant. Then we introduce a metal precursor, same like we do in our conventional methods, and then microorganisms then facilitate the reduction of these metal ions into metal nanoparticles. And this process can also be achieved using plant extract or plant biomass. With the various steps, I will explain it further in next slide, but not in too much detail. The magnified section in the here and here, it is showing the biological reduction of metal precursor. Microorganisms or plant extract essentially donate electrons to the metal ions, causing them to clump together to form nanoparticles. This is a natural, environmental friendly way to create these nanomaterials. And then the resulting biogenic nanoparticles possess several desirable properties. As you can see, they offer high surface area control. We can control the particle size and shape also, and then crystallinity, high stability, and also biocompatibility, low toxicity, and tunable functionality. These characteristics make them ideal for various applications. And application can be in biomedical applications such as drug delivery and diagnostics, or antimicrobial and antibacterial properties, environmental remediation for cleaning up pollutants, and agricultural application to enhance crop production and protection. This demonstrates a versatility of the potential of biogenic nanoparticles in creating sustainable technologies. So I will skip this slide. So this slide is showing the biogenic synthesis of nanoparticles. Like in this case, we isolated the bacteria from soil sample, and then soil bacteria culture was prepared, and then biomass was separated. Actually, there are two ways to synthesize using microbes. One is intracellular synthesis. That is, nanoparticles are synthesized inside the bodies of microorganisms, while the second one is extracellular synthesis. That is, nanoparticles are formed in outside the bodies of microorganisms. In that case, we use like as the microbes release metabolites, those metabolites can act as a bioreductant. So in this case, we have done extracellular synthesis for the nanoparticle formation. When zinc oxide precursor, zinc precursor was added, like zinc salt, either chloride or sulfate, and then this flask was incubated for 24 hours. We use different temperatures, and then biological reduction happened during that time span, and then we got like precipitate same as we got in the conventional synthesis method. But in case of biogenic synthesis, the color will not be same as the color of our nanoparticles which we produce. Like in case of zinc oxide nanoparticles, when we synthesize zinc oxide nanoparticles using conventional method, then the color is white. But you can see here at the bottom right side, the color of our nanopowder is like yellowish. Similarly, if we use plant extract, it depends on the color of plant. It will impart that particular color to our nanomaterial powder. And it is also an indication that the nanoparticles are functionalized, are coated with the various molecules present in the flask. That is that in this case, those are like metabolites. And then we write the sample and then send for characterization, and then we did its application study. So similarly, we same slides, but different material. We have done UV visible spectra. Then we also got FTIR, XID results, and then FISEN and HRT-EM results, and EPS, and also the particle size in this case is 14.5 nanometer average particle size. This average particle size we calculate from, like, we measure the sizes of various nanoparticles from various images. Let's say we choose statistically some sample range, let's say like 150 plus, like in my case, I will choose like that. And then we plot their histogram and we calculate their particle size. Like you can see that the majority of nanoparticles, the bars in the middle are less than 15 nanometer. Then we also have some nanoparticles more than 15 nanometers. And we also have some nanoparticles, but the frequency of those nanoparticles are lower. That's why the average particle size is 14.5. So we have done its antibacterial activity as well. And then in conclusion, we have successfully synthesized zinc oxide nanoparticles using sustainable biogenic approach with the soil bacteria. And these nanoparticles exhibit promising antibacterial activity. And this research contributes to the sustainable development goals by addressing the antimicrobial resistance and also to the SDG 9 by advancing the sustainable nanotechnology. So this is also one of work we have done, actually this work is in collaboration with Dr. Tamim from University Malaya. Before coming here, when I was in Malaysia, we have done this work in Malaysia, and then we have repeated the same work in our lab, and then like in this case, we have used the box sonicator, but here I improved the method and we used the probe sonicator, and we produced like nanocurcumin. Curcumin is the extract from the Curcuma longa L, which is known as turmeric. I'm sure you are familiar with this as like turmeric. So we use it in our daily life cooking. So we did like succinate extraction, and then the curcumin, we extracted the curcumin. Curcumin is actually mixture of different curcuminoids, curcuminide A, B, and C. We also did its TLC and other characterizations, which I didn't mention here due to shortage of time. I just explained the overview of it. So then we ultrasonicated it, and then we use some different techniques to synthesize nanoparticles, and we had done the antimicrobial susceptibility test. And then further extending this work, we have collaborated with Dr. Safri, who is from medical science, and she's helping us to study anti-cancer activities of these nanocurcumin, and also like the effect of the nanocurcumin with the on the SARS-CoV virus. Nanocurcumin is very interesting. I'm interested in these kind of materials because these are natural foods, but their bioavailability is more compared to like curcumin. The nanocurcumin has more bioavailability, and it is more soluble in water compared to curcumin, which is strangely soluble or almost insoluble in water. So now I move to the application part in nanobiotechnology and also in microbiology. Nanoparticles enable targeted solutions. For example, in drug delivery, they can be precisely target disease cells, minimizing the side effects. And in similar way, biosensors can become more sensitive, and tissue engineering benefit from improved performance. In the coming slides, I will discuss a few of them. Like, for example, drug delivery. Nanoparticles have transformed drug delivery systems by offering precision, efficiency, and targeted solutions. Here are some key applications like targeted drug delivery, and then crossing the biological barriers, and then controlled release, cancer therapy, combination therapies, and improved solubility. Actually, the nanoparticles, when they are in nano size, we can also functionalize them and tune or tailor, as I discussed earlier. We can tailor according to our application. Target application, then we can tune accordingly. Tune means we can functionalize nanoparticles. For example, if I synthesize a nanoparticle, and if I collaborate with any of the other field people, they, if I give them my nanomaterial, they will say, okay, we need oil soluble, but your nanomaterial is not oil soluble. Then what is the solution for that? We do the surface modifications for that. For example, we functionalize it with some organic molecules, which will I will use the like simple term like which will functionalize the nanoparticles. They can swim easily in the oil medium, so it becomes lipophilic. Similarly, if someone needs hydrophilic nanoparticles, we can use different functional groups to make it water soluble. That is hydrophilic. And then in some cases, we also need amphiphilic. That is both oil soluble and water soluble, and the nanomaterial can distribute between oil soluble oil and also in the water. So when we tune it, we can tune it according to our desired application, such as drug delivery. Yeah. So the next interesting application are biosensors. You know that biosensors are extensively used in different fields. So like medical diagnostic for detecting diseases, monitoring the glucose level, etc., etc. And then also for environmental monitoring, like detecting the pollutants or toxins or hazardous substances in the air, water, or soil samples. And then for food safety as well, like insurance for food quality by detecting the contaminants like bacteria or pesticides. Here I will also mention like uh, we can make nanoparticles and then we immobilize with some of DNA. Like for example, in my lab, one research group was working on nanobiotechnology. They were detecting like um, uh, detection of non-halal in processed food. Like if you see a burger, you cannot say that it is either halal or non-halal. And similarly, like for the Hindu people, they don't consume beef, so they cannot distinguish whether it's beef or not. But if we can make biosensors which can detect the particular DNA in the processed food, then it is very convenient for and we can use it. And it is, I think in UPM they have created they have made a kit which is like they call it, I think, halal kit to detect the non-halal content in the processed food. Similarly, it is also aiding us in the drug development, assisting the pharmaceutical research by analyzing molecular interactions. Then another crucial application is enhancing the enzyme production. By immobilizing enzymes on nanoparticles, we can significantly improve their stability and activity. And this has major implications for industrial process such as biodiesel production. Nanoparticles also they act as a support for the enzyme immobilization, and then it also enhances the stability and activity of the enzymes. And here is an example, like for example, in this figure, sorry, I didn't add citations due to shortage of time, but if you need, I can provide you the link for this paper also. Like in this case, this enzyme is functionalized or immobilized with the magnetite nanoparticles, which has NH2 group on it, and it was used to convert waste oil into biodiesel. Then nanoparticles in microbiology. In microbiology, nanoparticles are powerful tools against pathogens and biofilms. They can also aid in environmental cleanup and enhance agriculture practices, addressing the critical global challenges. So nanoparticles play a key role in bioremediation. They improve the efficiency of microorganisms used to degrade the pollutant, making them making environmental cleanup faster and more effective. So similarly, nanotechnology is also used in agriculture. Nanoparticles are used to improve the delivery of nutrients. For example, if we use like nanofertilizers, so it can improve the delivery of nutrients, and also as a nanopesticides. So this improves the overall health of crop yield. And other than that, we can also use them both but we also have toxicity concerns. So those biocompatible nanomaterials we can use as a coating for the different fruits and vegetables to enhance their shelf life. So this is one of the example of use of silver nanoparticles for antimicrobial coatings on medical devices. So this significantly reduces the risk of infection and benefiting the patient's safety. Recently, I was watching one BBC documentary that in Pakistan, there was a hospital in Multan, I guess. The people with the dialysis problem, like they go for with the kidney failure, they go for dialysis frequently. So many of them were infected with HIV virus, and the reason was like someone was HIV positive and he went for dialysis, and then the same equipment, medical equipment was used, and then I think more than 15 people were infected due to that. So if we use like a nanoparticle coating, which act as an antimicrobial coating, so it can reduce the risk of infection. During my PhD work was also we got inspiration from project, which was Hartaliga project. They wanted our research center to produce a magnetic cloth having antibacterial properties. So when researchers from our research center, they could produce like magnetic nanoparticles, but the problem with magnetic nanoparticles was like as they were using like iron oxide nanoparticles. Iron oxide nanoparticles can be easily oxidized, and with the passage of time, the magnetic properties will also change. So my PhD project inspiration was from that project, and then we started functionalizing magnetite nanoparticles with various antioxidants. So this slide highlights the interconnectedness of various biotechnological fields in building a sustainable bio-based economy or circular economy. By leveraging the nanotechnology and enzyme development, we can create innovative solutions for a wide range of industries, leading to a more environmental friendly and economically viable future. This is also this title, and this figure is from one of the book chapters. Maybe you can search the same title. You can see this is a book chapter from a book. Sorry, I didn't provide citations here. So now let's connect our research to the broader global context. The UN Sustainable Development Goals provide a roadmap for a better future. Nanotechnology and microbiology is playing a crucial role in achieving these goals, as well as biotechnology. So these are the SDG goals. So now our work directly support SDG 3. How? When we synthesize nanoparticles using environmentally friendly method, actually we can also use the nanoparticles synthesized by conventional methods. But the problem is same, like those are not environmental friendly methods. So if we can produce biocompatible nanoparticles with some kind of greener method, which is environmental friendly, it is much better. And then how our work directly supports SDG 3 is like advancing the healthcare solutions, nanoparticle-based drug delivery, antibacterial coating, biosensors, and all these applications are contributing to the better health outcomes. Then, biogenic nanoparticles, greener nanoparticles, they can also contribute to the clean water and sanitation, which is also SDG 6, one of the sustainable development goals. So clean water is essential for us. So our research on nanoparticles can enhance the like bioremediation and directly addressing the SDG 6 by providing a sustainable solution for the water purification. Then also it is interconnected to the SDG 12, which is responsible consumption and production. So we are committed to responsible consumption and production. So green synthesis and the use of biomaterials are key to minimizing our environmental footprints and promoting the sustainable practices. Then climate change has also a pressing issue. Our work is also can contribute to SDG 13 by developing solutions for environmental remediation and promoting sustainable agriculture. So today we have explored the fascinating history and diverse applications of nanomaterials, from ancient art to the modern biogenic synthesis and enzyme development. Nanotechnology, nanobiotechnology, and offers immense potential. These fields offer immense potential. Ultimately, the future of nanotechnology lies in its responsible development and applications. We must consider the ethical and environmental applications of our work, ensuring that these powerful tools are used to benefit society and planet. Together, we can unlock their future potential and create brighter future for all. So this is my output progress in the BRIN last 1 year. This is my last month. I will be leaving at the end of this month. So far we have produced data for 4 papers, and then one of our paper is accepted in Q1 journal, and then we also filed 1 patent, and I also actively participated in writing research proposals, and we submitted 2 research proposals this year, but we could only got acceptance for 1. So this is our accepted manuscript, which is the title is Nature Nanofactory, Biogenic Synthesis of Non-Metal Nanoparticles for Sustainable Technologies. It is published in Green Chemistry Letters and Reviews. The impact factor is 5.8, and it is highly reputable journal. This is our second manuscript, which I also presented today. It is a zinc oxide nanoparticle synthesis and characterization and their therapeutic potential. We have submitted this work to the nanomedicine, nanotechnology, biology, and medicine. Impact factor is 4.2, and we hope it will be published soon. Then the third manuscript, which I also mentioned in one of our slides, nanocurcumin in graphene nanoplatelets hybrid, a promising composite for antibacterial properties. We have submitted to World Journal of Microbiology and Biotechnology. Impact factor is 4, and all these journals are highly reputable. And our fourth journal, manuscript is ready for submission. The title is Biogenic Synthesis of Metal Oxide Nanoparticles with Enhanced Antibacterial Properties. So during my stay in the BRIN, I have tried my level best to collaborate with other international collaborators. Like first of all, we have collaborated with my home university, Nanotechnology and Catalysis Research Institute, University of Malaya, Kuala Lumpur. And then we also collaborated with Mechanical Engineering Department of Yonsei University, South Korea, and Mechatronics Engineering in the Atlantic Technological University, Sildo, Ireland, and also with the King Khalid University, Abaha, Saudi Arabia, and also with Bahauddin Zakriya University, Multan, Pakistan. So these are some of my knowledge sharing. This one, which is happening now, sustainable innovation, harnessing nanoparticles for nanobiotechnology and microbiology. And last week, we also attended JAI's roles towards the adaptive, resilient, and sustainable innovation Indonesia. So other than that, I have attended like 3 days workshop on applications of in biology, and also attended 1 day workshop on writing papers. And then I also attended several webinars on various topics. And some of my starting slides, you might have noticed that I focused on the intersection of science and art. Actually, these are the review papers which I produced. These are not published yet, but we have completed, and 2 of them are submitted. But as it will not be counted as my KPI, but I have submitted 2 of them. One is Evolution of Nano Art and Its Future, Ideal Union of Creativity and Science. And the second one is Nano Art, Aesthetics in Nano Engineered Materials. The third one is Nano Canvas, the artistic expression in the age of nanotechnology. And the fourth one is Flora of the Nanoscale, artistic nanoflowers. So when we see the nanomaterials under the microscope, sometimes we see very interesting structures. Like, for example, nanoflowers. We can produce many kind of nanoflowers. Although, as a scientist, we are more interested in their applications, like for my biomedical or engineering applications. But this one is also like everyone like to see like beautiful, and everyone have aesthetic sense up to some extent. So I perceive them as when I see the nanomaterials under the microscope, electron microscope. So then it depends on all our imagination how we see it, and then we can color the image from the SEM. As you know that those images are not colored. Those are just like grayscale or black and white images. So we can just color them and create a nice piece of art, as our ancestors used to do. Like I gave example of cave paintings and also the earlier civilization. I didn't mention here about Mayan civilization and some other details of Chinese, how Chinese use the nanoparticles, because due to shortage of time. But in these review papers, I have explained them in more details. Thank you very much for your attention. I'm now happy to answer any question and you may have.
Okay, thank you very much, Dr. Tawab, for that insightful presentation. We appreciate your sharing your expertise with us today, from the history of nanoparticles, the characterization, the application for any field such as biomedical, biosensor, microbiology, bioremediation, and also in enzyme productions as a biocatalyst, and the relation with SDG, and also the many achievement as long as Dr. Tawab collaborate with BRIN as a postdoc, almost 1 year. Now, still only 46 participants. I would like to open the floor for questions. If you have any inquiries or talks regarding the presentation, please feel free to raise your hand and type your question in English or in Indonesia in the chat. Dr. Tawab will be happy to answer it. Now there is a question from Dr. Sulistiani. In Indonesia, apakah nanopartikel ini bisa untuk enkapsulasi bakteria? The question is it can be the nanoparticle as bacteria encapsulation? And is there any evidence about it?
To the best of my knowledge, we do encapsulation of drug loading of our drug loading on like for example for the targeted drug delivery, but I believe it is possible now. Like same as like we load the drug on the nanoparticles for targeted drug delivery. Similarly, any microbes can be encapsulated as well.
If Bu Sulistiani not enough, so please can open your mic and ask now. Or maybe how about the size, Doctor, between nanoparticle and the bacteria?
Yeah, that depends on the synthesis method. For example, today I showed in my slides like the conventional nanoparticles we have synthesized have nanoparticle size of less than 5 nanometer, while the result of biogenics which I presented today, the particle size is 15 nanometer, almost less than 15 nanometer, roughly 15 nanometer. That depends on our synthesis method. Actually, here I didn't mention about drawbacks or disadvantages of our biogenic methods. Like one of the main challenge for biogenic synthesis methods are like those we can sometimes we cannot scale up. Like for example, on lab scale we can synthesize the nanoparticles using like sterile condition and very sophisticated, especially in case of bacteria and fungi. We need very clean environment. While when we scale up the process, we may have we may face the problem for that. And the second is also like it depends on the bacteria. Like how it depends on bacteria, because different bacteria produce different metabolites. So depending on the concentration and also depending on the nature of the metabolites, which act as a bioreductant, the size can vary. Like for some studies, you will see like nanoparticle size is 50 nanometer, but same nanoparticles like, for example, zinc oxide, and also shape. Shape also depends. Like, for example, if you are using bacillus, any bacillus bacteria, maybe you will get like spherical shape. But if you change the bacterial species, maybe you will get like a rod shaped nanoparticles, and similarly different size as well.
Okay, Doctor. Still with us, Dr. Tawab? Hello, Dr. Tawab.
Yes.
Okay. Okay. Any question from other participants or Ibu Sulistiani have? Oh, there is another question from Bu. Wait. Okay, enough. The next, the next, the next question is from Ibu Apridah Kamriawati. Is it possible if we want to reforming the enzymes such as cellulase, silanase, and many others into nanoparticle size? What kind of method we will use, and what kind of parameter that can be the priority for getting optimum result if you want to make nanoparticle those enzymes? Thank you for your explanation.
Okay. It's very interesting question. Like how we can make the enzyme is like in a nano form, like I explained about the nano curcumin. So there are various methods that can be utilized. I'm not sure about this enzyme particularly, like cellulase and cellulase, sorry, but I'm sure there are techniques which or maybe we can discuss in more details if you explain to me more about or if I study their chemical nature, then I can propose, like for example, communication is one of the methods. We can reduce the particle size of our this one. And similarly, if you have bulk production of your material, then maybe you can use ball milling as well to get the particle size of your material to the nanoscale. That is also that is a physical method, not a chemical method. I hope you understand it.
Oh, but Dr. Afrida, are you satisfied with the answer? So it is a possible, doctor, to get a nanoparticle from enzyme into.
Yeah, it depends on their chemistry actually.
Oh, depend on the chemistry.
Yeah. Chemistry and other physical properties as well.
And the physical property.
Yeah.
All right. Okay, thank you. If any question, you can write on the chat or directly. Okay, we're waiting for another question. I have one question, Dr. Tawab. You mentioned about the related between nanoparticle and SDGs. Actually, our country Indonesia have a focus on food security. Can you explain more how to nanoparticle, nanomaterial can scale up for the for our food security in Indonesia?
Yeah. First, first of all, this is also a very interesting question. Thank you for such an interesting question. Actually, the crop production, we can enhance the crop production by using nanoparticles. But whenever it comes to the nanoparticles, then there is also concern about nanotoxicity. So if we produce the nanoparticles using such a sustainable way, so it can also increase our crop yield, which can end up with the like to eradicate those problems like less crop yield, etc. And similarly, as I mentioned, like if we use like nanopesticides and nanofertilizers, it can enhance our crop yield. And another is like interesting and important thing is responsible consumption in production, as we said.
Okay, doctor. Any addition, doctor?
No, I think.
Okay, thank you for suggestion for the relate for the food security. And then there is a question from Dr. Dadang. He is a lecturer from University of Sudirman in Purwokerto. Nice talk, Dr. Tawab. I have a question. How about the toxicity of the zinc oxide if administered to human or animal? Please.
Okay. So far, the zinc oxide is considered as a less toxic, or you can say like biocompatible, but concerns are there. As Dr. Badang highlighted, the concerns are still there, but mostly the zinc oxide is used our skincare products like creams, which especially the ladies are using almost every day. So zinc oxide is one of the component, and then zinc oxide is also used as a coating for the fruits to enhance their shelf life. So there are less concerns compared to other nanoparticles. And one way is to reduce the toxicity is like when we use the conventional methods, those are like more, it is not directly toxic. We cannot say that this is nanotoxicity, but we are generating like more waste. So that's why we are looking for the sustainable methods. I respected a bit from it, but I think it is important to mention this. So nanoparticles, there are always concern about when it comes to the nano particles, and this also depends on the particle size. Like if particle size is less than for like drug delivery and also for like topical application, normally the suggested one is more than 100 in between 200 to 200 nanometers. When the particle size is less than that, then there are concerns about their nanotoxicity. So similarly, in other applications as well, like if you have particle size 5, then the toxicity is different. If the particle size is 10, the toxicity is different, and then 50, the toxicity is different. So there are still like I remember one quote, they say that science is always wrong. It creates it solves a problem by creating 10 more. Actually, this is a research in research field we also encounter. Like in chemistry, we had many accidental discoveries, and then like for example, chlorofluorocarbons. When it was discovered, then people calling it like magical gases. They were extensively used in the refrigerators, air conditioners, and then later on research showed that those are main culprit for the ozone layer depletion. So when that's why I put this slide, responsible consumption and production. So and similarly, during my PhD work, we have synthesized and functionalized magnetite nanoparticles. So because my first step was to synthesize multipotent antioxidants. And also I use like a natural antioxidants, and I choose gallic acid as a water soluble, and then quercetin as lipophilic. So before synthesizing those multipotent antioxidants, we have done like past predictions, which can predict the biological activities of the organic molecules. And then we have done like computational studies, structure activity relationship, and also admix studies to make sure that our organic molecule which we are going to synthesize is has a good safety profile. And before synthesis, we did those computational studies. So similarly, I think we should also think about toxicity concerns uh, before we start to synthesize or scale up the procedures or process.
Okay, thank you very much, Dr. Tawab. Thanks for Dr. Dedang. And now the fifth question from Dr. Aprida. Dr. Tawab, enzymes is a sensitive protein. If we reform it become nanomaterial, is there any possibility it can reduce the enzyme activity? Is there any suggestion to avoid that matter?
Can you please repeat the question? Sorry.
Okay, enzymes is a sensitive protein.
Yeah.
If we reform it become nanomaterial, is there any possibility it can reduce the enzyme activity? Is there any suggestion to avoid that method?
Yeah, because enzymes are proteins, so definitely that is very challenging work to convert them into nanoparticles. But I believe that the best way is to immobilize it with the nano materials, nanoparticles. So, which is a best practice, which mostly the people are doing to avoid those kind of like, instead of thinking about difficult work, they are just immobilizing, like I mentioned in one of our slides, that functionalized, they used functionalized magnetite nanoparticles to immobilize the enzyme, and then they used it for the production of waste oil into biodiesel. So enzyme activity can be enhanced. So the best alternative way is to immobilize it, and the easy way is to immobilize it with the nano materials.
Okay, thank you, Dr. Tawab. As described before, that we can immobilize the enzyme, yeah, with nanoparticle to enhance the enzyme activity. Okay, for any application. Okay, any addition? Question from Ibu Afrida or from Dr. Tawab for. Okay, enough, Dr. Afrida. Okay, if any question from other participant, no, still 40, more than 40 participant here. I think it's enough. Okay, Dr. Tawab, do you have any close statement or anything in this session?
No, thank you so much for giving me this platform to share my knowledge with the BRIN community. And I really enjoyed my stay in Indonesia, and I learned many Indonesian words, like, as I know some of the basic Bahasa Melayu, but there are several different words I like, which were new for me. And then I learned Bahasa Indonesia sedikit-sedikit, and I will continue learning it sedikit-sedikit jadi bukit. So, and also I enjoyed the diverse cuisine of our country, Indonesia. And I really enjoyed working with the Indonesian people in my lab, and also other than our lab, wherever I went, I was treated very well and treated very well. So thank you so much. That's all from my end. And special thanks to Dr. Ira and Prof. Devi also, who put their efforts to bring me here from Pakistan and gave me this opportunity.
Thank you very much, Dr. Tawab. We appreciate you sharing your expertise with us today from A to Z and very, very useful for us and make open mind for me especially. And thank you for ladies and gentlemen, Bapak Ibu semua for attending in this session and many question. We hope we can see you later. Thank you very much. Thank you, Ibu. Saya. So the session to the MC.
Okay, thank you, Dr. Ira, for leading a very interactive discussion, and thank you, Dr. Tawab, for a very interesting presentation. And as a token of our appreciation, we will give a certificate to Dr. Syed Tawab Syah as a speaker. Thank you very much for sharing your valuable knowledge.
You are welcome.
Okay, ladies and gentlemen, this is the end of our agenda for today. I'm Andalusia Arini. On behalf of the committee, I would like to say thank you very much to our speaker, moderator, and all participants. And if you want to know more about Research Center for Applied Microbiology BRIN, you can check our social media on Facebook, Pusat Riset Mikrobiologi Terapan, and Instagram at rmt_BRIN. You can check the information of the webinar speaker on the Instagram story highlights, and you can also check the recorded webinar on our YouTube channel at PR Mikrobiologi Terapan. See you in the next event. Happy fasting for those who are observing it. Wassalamualaikum warahmatullahi wabarakatuh.
Terima kasih Mbak Ira, Mbak Uci. Thank you, Dr. Syed Tawab.
You are
Terima kasih Bapak
welcome.
Ibu semuanya.
Thank you, Dr. Tawab.
Maaf lahir batin kalau ada salah.
Mbak Ira, makasih Mbak Ira.
Iya, sama-sama
Okay.
Mbak.
Okay.
Mbak Uci, makasih.
Terima kasih Mbak Uci, Mbak Prilia.